Hello, everyone. First of all, I would like to thank the organizing committee for giving me this opportunity to share my work. I really wish I could be with you all, but I am awaiting the arrival of a little future prion researcher, and so I'm joining virtually instead. And I'm very excited to share my work on spontaneous prion emergence and knock-in mouse models. So, as we all know that prion diseases, they are rare, fatal, and incurable brain disorder caused by prions. And I like to refer prions as protease-resistant, infectious, organ-specific, neurotoxic, self-replicating proteins. In short, these are like rogue proteins which misfold, spread in the brain, and just take over. So most of what we know about prion disease work comes from the classical model, which involves infecting animals with the preformed prions. And this has been incredibly useful in the field to study late onset disease, but it does not reflect how prion diseases start in most people. In fact, less than 1% of human cases like Puru or iatrogenic CJD, these are arise due to infection. And vast majority of the diseases, like 99% of them, they arise spontaneously, either sporadically or due to the presence of disease-associated mutations like E200K or D178N, which kind of speed up the process. So the main challenge here is that spontaneous onset is rare, it's unpredictable, and very hard to model. And rather more, the existing current models, most of them, they do not replicate this situation very well. So the main question here is that how prion disease begins without infection and how we can model and ultimately intervene in the process early so that we can make a difference. So we began exploring the spontaneous prion disease by designing these knock-in mouse models expressing bank hole PRP. Now, you all might wonder why bank holes or what is so special about bank hole PRP. Now, these bank holes are considered to be universal acceptor as they are highly susceptible to different prion types. And unlike most species like hamsters or mouse, which are commonly used as the animal models, these do not offer strong species barrier, which means if you have wild type bank holes or even transgenic mice expressing bank hole PRP, and they will get sick and develop the disease spontaneously. So, in collaboration with Walker Jackson, we designed these knock in mouse models, which were expressing physiological levels of bank hole PRP wild type protein and bank hole PRP with E200K and D178N mutations, which are linked with fetal familial insomnia and familial CJD, respectively. So, here's what we found that the knock in mice, which were expressing mutant bank hole PRP and not the wild type versions, they developed neurological illness over time and took like 400, 500 days of age and showed range of symptoms like hunched back, tremor, bradykinesia, weight loss, and some skin issues like dermatitis. And we then took a look at the brains of these sick mice, and we found that these has prions, which were resistant to common proteases used in the field like thermolysin and proteinase K. And interestingly, we also found that upon digesting with proteinase K, they showed these atypical fragments which were at 10 kilodalton. Now, we call them atypical because these are not typically seen in the patients carrying E200K or D178N mutations. And we also looked at the brains of the sick mice, and they showed these neuropathological changes. We found these tiny holes in the brain, which we called as vacuolation, and extensive brain damage and neuroinflammation, which is typically related with the prion diseases. So last year, we published this whole study in Journal of Clinical Investigation. So if you are interested in learning more about it, you can always refer back to the article for the more details. So now the knock-in mice that we generated here, they were homozygous, which means they were having two copies of the mutant gene. But in actual, like 99% of cases of human genetic disorders, these are heterozygous, which means they carry one copy of wild type and one copy of mutant gene. So what we did in order to mimic the human cases and in order to understand the effect of gene dosage that it can have on the spontaneous misfolding, we created these knock-in mice heterozygous, which means they were having one copy of the wild type gene and one copy of the mutant gene. So, in addition to that, we also created these compound heterozygous mice, which were having both the copies of like different mutations in the same host, so that we can study if there is any interaction that can happen in the same host when you have two different mutations. So far, here's what we found that the knock-in heterozygous mice, which had one copy of the mutant, they did get sick, but more slowly than their homozygous counterparts. And on the other hand, the compound heterozygous mice, which had both the copies like one E200K and one D178, and they got sick just as fast or a bit faster than their homozygous counterparts. And all these mice showed similar like range of symptoms like tremor, bradykinesia, weight loss, some limb abnormalities, and dermatitis. And the brains of the sick mice, they showed the protease resistant prions, and we also found these atypical PRP fragments upon digesting them with proteinase K, which were earlier present in the homozygous mice as well. So we are currently analyzing more and more of these brains so that we can test them for the brain damage, any neuropathological changes and prion related changes, the seeding activity, which can tell how these prions are spreading. The thing is that what we believe, so in our data, we were getting these atypical PRP species, and there was no difference between the E200K and the D178N mutations in our knock-in mice models, right? So, but in actual, people with E200K and D178, and they are two different diseases, and you get to see the symptom differences in the disease profile. Now, so far, our data was reflected only the first step of the mechanism. So it's just like in the, like mice, they have the age limit, like you cannot incubate them for more than 2.5 years, right? So we were able to just show the first step, like when the normal prion protein was getting converted into these atypical PRPSC. And now we believe that in humans, what happens, first step, like in the early misfolding, these atypical species are formed. And later on, in the later stage of life, which takes like years to happen, these atypical undergo further conversion and form these classical PRP species. And this step is now when the mutations kick in, they play a role, and the symptoms, like in actual human cases, they get differences in the symptoms and the disease profile differs. So far, we have only modeled the first step. And the second step, we are still working on it. And in future research involves this atypical to classical conversion we can show in the knock-in mice models. So in summary, what we found that the knock-in mice, which were carrying these human CJD and FFI-related mutations, they developed neurological illness on their own without any exposure to infectious prion. They showed range of symptoms as I discussed in my previous slide. Now, in our current study, we did not find any specific differences between the two mutations in the disease profile, which suggests that the common prion formation, which occurs across mutations, is same, or they are resulting in the similar type of strain, regardless of the specific mutation involved. And the third very important point is that gene dosage matters. In R, we found that the heterozygous mice, they did get sick, but much more slowly than the homozygous counterparts, which means the people with the two copies of mutation, they may be at a greater risk and they develop the disease at earlier stages compared to their heterozygous counterparts. So in future, we we are trying to explore how these different genetic mutations, they can cause prion proteins to misfold in a specific way. Because we know that human cases with G1s, with E200K and T178, and they have different disease profile and different neuropathological changes. So what we are trying to understand is that in our models, how these genetic mutations can cause the proteins to misfold in certain ways. And that we would like to understand by studying these atypical forms and how these atypical forms of the misfolded proteins can change into the more harmful, the classical ones that we typically see in the patients in the full blown disease with E200K and D178N mutations. And last but not the least, we are trying that we could target those atypical form, because if we target those atypical form, we can completely stop the misfolding process early on and block the conversion of the atypical into the classical prion versions from forming all together. So we can act early. So if you have any questions, feel free to reach me out via my email address, or you could even connect me on LinkedIn. And if you are interested to learn more about these knock-in mice, you can scan the QR code to read the full article. And to conclude, I would like to thank the entire Watts Lab, and special thanks to Joel Watts for being an amazing mentor, and Erica Stewart for handling and taking care of all the mice. And last but not the least, I am sincerely thankful to all the donors and funders for their generous support and contribution, which has made this research really, truly possible. Thank you so much.